An integrin molecule consists of a large alpha chain that pairs non-covalently with a smaller beta chain. There are several subfamilies of integrins broadly defined by their common beta chains. We will be concerned chiefly with the leukocyte integrins, which have a common beta-2 chain with distinct alpha chains. All T cells express a beta-2 integrin known as lymphocyte function-associated antigen 1, or LFA-1. This leukocyte integrin is also found on macrophages and neutrophils and is involved in the recruitment to sites of infection. LFA-1 plays a similar role in the migration of both naive and effector T cells out of the blood. In addition, it is thought to be the most important adhesion molecule for T lymphocyte activation because antibodies to LFA-1 effectively inhibit the activation of both naive and armed effector T cells. Surprisingly, T cell responses can be normal in patients lacking the beta-2 integrin chain, and hence all integrins that contain beta-2, such as LFA-1. This is probably because T cells also express other adhesion molecules, including CD2 and beta-1 integrins, which may be able to compensate for the absence of LFA-1. Expression of the beta-1 integrins increases significantly at a late stage in T cell activation, and they are thus called VLAs, for very late activation antigens. They play an important part in directing armed effector T cells to their inflamed target tissues. Many cell surface adhesion molecules are members of the immunoglobulin superfamily, which also includes the antigen receptors of T and B cells, the co-receptors CD4, CD8, and CD19, and the invariant domains of MHC molecules. At least 5 adhesion molecules of the immunoglobulin superfamily are especially important in T cell activation. 3 very similar intercellular adhesion molecules, ICAMs, ICAM1, ICAM2, and ICAM3, all bind to the T cell integrin LFA-1. ICAM1 and ICAM2 are expressed on endothelium as well as on antigen-presenting cells. Binding to these molecules enables lymphocytes to migrate through blood vessel walls. ICAM3 is expressed only on leukocytes and is thought to play an important part in adhesion between T cells and antigen-presenting cells, particularly dendritic cells. In addition to binding LFA-1, ICAM3 binds with high affinity to a recently discovered lectin called DCSIGN, which is found only on dendritic cells. Another interaction involving immunoglobulin superfamily molecules is mediated by LFA-3 on the antigen-presenting cell binding to CD2 on the T cell. This interaction synergizes with that between LFA-1 and ICAM1 and ICAM2. Have a great day.